Hello, my name is Karlsen-Ayala, and I'm a PhD student at the University of Florida with Dr. Romina Gazis and Dr. Matthew Smith. Today I will talk about my work characterizing the spore bank associated with the Florida slash pines in the endangered pine rockland ecosystem. The South Florida Pine Rockland is a unique ecosystem only found in South Florida and the Florida Keys. Similar ecosystems are found in the Bahamas, Cuba, and Turks and Caicos, but the plant composition is slightly different. This ecosystem contains a mix of temperate and tropical plant communities. The pine rocklands are characterized by an overstory of slash pine trees, Pinus eleodii variety densa, and an understory of herbaceous plants and palms. Aptly named, the pine rocklands grow on an oolitic limestone substrate and require fire to maintain overgrowth of woody and invasive plants. As seen in this photo, less than 2% of the natural habitat outside of the Everglades National Park remains. This habitat destruction and fragmentation is mostly due to development. Additionally, invasive species and sea level rise also threaten this ecosystem. Therefore, this habitat is critically endangered and an important ecosystem to study for restoration efforts. While there are ample restoration programs in South Florida, as seen in this picture here, our lack of understanding of below-ground interactions may be a major hurdle in the impact or efficacy of these restoration efforts, particularly for pine trees. Pine trees are highly dependent on ectomycorrhizal fungal mutualists that provide nutrients to the plant in exchange for carbon. For many pine species, a lack of mutualists can prevent the establishment or persistence of pines. While the diversity of ECM fungi in other pine systems is well known, currently no comprehensive work has characterized the ECM fungi in the pine rocklands. Because this ecosystem contains several disturbances such as fire, we hypothesize that spore bank fungi are important for seedling establishment in this harsh ecosystem. Spore bank fungi are metabolically inactive spores that serve as primary inoculum for seedlings post-disturbance. These fungi tend to survive a long time in the soil and disperse easily. Therefore, our goals in this study were to determine which fungi are important in the spore bank, determine if there were differences between the fungi recovered from the spore bank, and determine if the soil communities were different across different habitats. We collected soil from sites across Florida in 3 different habitat types: sandhill, scrub, and pine rockland. This soil was sequenced with Illumina-MySeq using ITS1F and ITS2 to determine the total pool of fungi in the soil. The soil was dried for several weeks and pine seedlings were planted for 7 months. Then the pine roots were harvested and root tips were Sanger sequenced with ITS1F and ITS4. When we look at the fungi recovered from the spore bank on the seedling root tips, we see that there are no major differences in these fungi. Rhizopogon is predominant across all habitat types, and less common fungi in the spore bank varied slightly between habitat types. Other dominant fungi recovered on the root tips include Cenococcum, Thalopthera, and Psiloelis. This suggests that spore bank communities in the rocklands might not be that different from other habitat types. Interestingly, though, when we look at the total fungal communities in the soil, we see in this NMDS plot that the soil communities are pretty unique from each other, with samples from the same habitat clustering closely together. This suggests that even though the spore bank fungi on the seedlings are similar, other fungi in the soil are unique by habitat. In conclusion, this work serves as our first look at the below-ground interactions in this imperiled ecosystem. We've learned that rhizopogon are important in spore banks across multiple habitats, but the total fungal communities in the soil vary by habitat type. Future work should include ECM fungi from the mature community to better understand these differences in habitat type. This work will help inform conservation initiatives in this imperiled ecosystem. Finally, I would like to thank my collaborators, Dr. Gazis, Dr. Husino, and Dr. Smith, for their contribution for this project. Thank you.